Good morning, good afternoon, and good evening to everyone joining us from wherever you are around the world. Distinguished guests, ladies and gentlemen, it is my great honor to welcome you all to this highly anticipated awareness seminar. Before we begin with our main presentation, allow me to provide a brief overview of the initiative that brings us together today. INSTAL Africa, Innovation, Science and Technology for Action and Learning in Africa, is the result of collaborative efforts initiated in October 2023. This initiative leverages the Drosophila melanogaster fly model and state-of-the-art infrastructure to enhance fundamental research capacity in Rwanda. Rwanda has made remarkable strides in advancing its scientific capabilities, recognizing the pivotal role of research in sustainable development and public health. INSTAL Africa was born from a collaboration between the Foresight Institute of Research and Translation Rwanda, or FIRAT Rwanda, and its technical partner, Drosophila UK, in conjunction with key national institutions such as the National Council for Science and Technology, Rwanda Biomedical Center, the National Industrial Research and Development Agency, the University of Rwanda, and the Rwanda Food and Drug Authority. This collaboration embarks on a 5-year comprehensive capacity development plan aimed at cultivating the next generation of scientists and fostering research excellence across medical, applied, and fundamental sciences. From healthcare innovation to agricultural production, this partnership envisions driving progress in public health and sustainable development across Rwanda and beyond. It is against this background that we convene today, united in our shared commitment to advancing science, technology, and public health for the benefit of our communities. With that foundation laid, it is now my distinct pleasure to introduce our invited guest speaker.
Today, we have the distinct pleasure of introducing a scientist whose journey spans continents, disciplines, and an unwavering commitment to advancing laboratory medicine, Dr. Ridwan Ibrahim. Dr. Ibrahim is a clinical biochemist with Alberta Precision Laboratories in Calgary, Alberta, where he plays a central role in delivering high-quality laboratory services across one of Canada's major healthcare systems. His path to this role reflects both depth and breadth of expertise. Most recently, he completed a COMAC-accredited clinical chemistry fellowship at Baylor College of Medicine and Texas Children's Hospital in Houston. There, he stepped into the role of acting assistant medical director, supporting major aspects of laboratory operations, from method validation and quality assurance to the interpretation of complex diagnostic assays in one of the world's busiest pediatric settings. His academic and professional journey covers 3 continents. Dr. Ibrahim began his scientific training in Nigeria, earning both his BSc and MSc in anatomy from the University of Ilorin. He went on to complete a PhD in interdisciplinary neuroscience at National Yang Ming University in Taipei, Taiwan. Postdoctoral work followed at the Institute of Biomedical Sciences, Academia Sinica, and later in the Department of Clinical Neurosciences at the University of Calgary. This rich background allows him to blend rigorous neuroscience research with clinically focused laboratory medicine, especially in amyloidosis, neurodegeneration, and biomarker development. During his fellowship, he was deeply involved in interpreting some of the most complex tests in pediatric diagnostics: protein electrophoresis, alpha-1 antitrypsin phenotyping, CSF oligoclonal banding, von Willebrand multimers, plasma amino acids, and hemoglobin profiles, among others. He took part not only in testing but in ensuring quality, teaching residents and advising clinicians to optimize test utilization and strengthen patient care. His research program stands at the intersection of clinical chemistry, pediatric laboratory medicine, and translational neuroscience. He has earned competitive funding, including the ADLM Critical and Point-of-Care Testing Research Grant and the prestigious Pfizer Global Aspire Transthyretin Amyloidosis Research Grant. His work spans topics ranging from complement biomarkers in children to hemolysis detection at the point of care to machine learning approaches for distinguishing bacterial from viral infections. He has also been part of the growing conversation about the use of large language models such as ChatGPT in laboratory medicine. His publication record includes contributions to cellular and molecular life sciences, frontiers in physiology, clinical chemistry and laboratory medicine, annals of clinical and laboratory science, and many other high-impact journals. His earlier studies in Nigeria and Taiwan explored molecular mechanisms of amyloidosis, neuroprotective strategies in toxic and metabolic injury, and the pathology of environmental exposures, all of which laid a foundation for his current translational focus. His excellence has been widely recognized. Among his honors are the ADLM Texas Section Outstanding Abstract Award, multiple Society for Young Clinical Laboratorians Impact and Travel Awards, the University of Calgary's Harley Hotchkiss Samuel Weiss Postdoctoral Award, the Taiwan International Graduate Program Doctoral Fellowship, and numerous international awards from organizations including AACC, IBRO, and ISN. Beyond the laboratory, Dr. Ibrahim is a devoted educator and mentor. He previously served as assistant lecturer in human anatomy at Kampala International University in Uganda, teaching anatomy, embryology and histology, and guiding both undergraduate and graduate students. He continues to mentor young scientists through SciComm Nigeria, the Canadian Black Scientist Network, and earlier through Nigeria's National Agency for the Control of AIDS. Taken together, Dr. Ridwan Ibrahim represents the model of a modern clinical biochemist, internationally trained, research active, clinically engaged, and deeply committed to mentorship, equity, and the advancement of laboratory science for better patient outcomes. It is truly an honor to welcome him to our webinar today. Dr. Ibrahim, the floor is yours.
Good morning, all, or good evening, wherever you're aiming. Thank you so much for the kind introduction and for the invitation to present my work. I hope everyone can hear me. Okay, good. Thank you so much. So today, um, the title of my talk is The Curious Scientist Path from Amyloid Aggregates to Analyzer. So it's not going to be like a formal talk. I kind of put together a story of my journey, and my aim is to spark interest in young people and to also enlighten us about other parts apart from research. So feel free at the end of the talk to ask any question, and I'll be happy to provide some of the answers I have. So at the end of the talk today, I'll explain what amyloidosis are, and I will unravel the cellular mechanism that predicts early versus late onset tyrosine amyloidosis. Then after the whole biomedical research talk, I'll introduce PhD clinical fellowships, and I'll give tips on the application processes. Amyloidosis, these are protein disorders that are caused by misfolded protein deposition. These diseases are characterized by deposits and buildup of abnormal protein fibrils known as amyloid fibrils in tissues. When you stain an amyloid using Congo red, you have a green gold by vibrant color like this one shown behind. And amyloid protein can deposit in different organs with the eye as one of the organs that is being deposited in. Now, transthyretin is one of the proteins that's misfold. And this protein is a secretory protein. It is produced in the liver, the choroid plexus, and retina. And the function of this protein is to transport thyroxine and retinal binding protein in the body. So this protein is one of the major proteins that can also unfold. And when it unfolds, it leads to a disease called transthyretin amyloidosis. Now, transthyretin amyloidosis, like I said, is due to the unfolding of the transthyretin protein. So we have two types of transthyretin amyloidosis. We have the wild type amyloidosis and we have the hereditary amyloidosis. The wild type amyloidosis is due to the unfolding of the wild type protein. There's no mutation in the gene. The wild type protein just simply unfolds into fibrils and deposits in the body, majorly in the heart. But for the hereditary amyloidosis, this is due to a mutation in the TTR gene. And when there's a mutation in the gene, the mutation predisposes the protein to unfold. And this can cause either the familial amyloid polyneuropathy or familial amyloid cardiomyopathy. Now, this hereditary amyloidosis is as focused because we have two types of it. We have the early onset, in which the individual has disease as early as late 20s, early 30s. And we have the late onset, in which the individual have the disease in the late time of life, maybe around 50 and above. Now, how does this disease take place? Like I said, the transthyretin protein is produced by the liver majorly. Also, the choroid plexus also produces it. So the protein is released into the bloodstream. Now, when it's being released into the bloodstream, depending on the mutation or the wild type, and as the case may be, this protein, which is in tetramer, will unfold into individual monomers. Now, the individual monomers will now undergo modification and aggregate, and they aggregate as fibrils, and they deposit on the nerves, in kidneys, in the heart, in skin, and other different organs. So the unfolding of the tetramer into monomer is the rate limiting step in the disease pathophysiology. Now, the question we have is, we have over 100 different mutations on this gene. Now, how does the body knows which mutation will cause early onset and which mutation will cause late onset amyloidosis? So that was the question we asked, that which cellular mechanism predicts which mutation cause early or late onset ATTR amyloidosis. Now, before I did, so this was my PhD work. So before I did the work, there was a model that was available in the field. Now, basically, in the endoplasmic reticulum, we have unfolded TTR monomers. When there's a mutation, and these mutations are very unstable, the body does not secrete this TTR that are very unstable. It undergoes what we call endoplasmic assisted degradation. So this mutation will be degraded. They don't get secreted. But if the mutations are not very unstable, then in the ER, there's what we call the endoplasmic assisted folding. So the body will try to fold this unstable mutation. They are not very unstable. It's fold them into monomers, and also they get into tetramers. Now, from the endoplasmic reticulum, this protein are being packaged and moved into the Golgi apparatus. From the Golgi apparatus, they have some modification that is being done. Then they get secreted out into the bloodstream. Now, this is where the model says that when they now get secreted into this bloodstream, if the tetramer is stable, it remains stable and it carries out its function. But If the tetramer is unstable, then the tetramer unfold into monomers, and this monomer become unfolded monomers, and they later become amyloid. So the stability of the tetramer predicts the early onset or late onset. So what this model is telling us is if the mutation is stable, then the protein is stable and it doesn't unfold quickly. But if the mutation is very unstable, then it unfolds into monomers and become aggregates, which later deposits into the blood and causes the disease. Now, when I undertook this project, our question is, this model is not enough to predict the onset of amyloid amyloidosis. So to tackle this question, our question is, what mechanism predicts this onset of disease? To tackle this question, we focus on the Taiwanese mutation. There's a Taiwanese mutation of this disease that is called the A97S. This mutation is unique to Taiwanese, and you have a change from alanine to serine at amino acid 97. Now, this particular mutation is a late onset mutation, and when patients have it, it's a very aggressive mutation, and the patient actually die within 6 to 8 years of the disease onset. So we're trying to use this mutation to understand how the body predicts which mutation will be early onset and which mutation will be late onset. So we had two hypotheses. Our first hypothesis is that this A97S mutation, which is a late onset disease causing mutation, is stable. So because it is stable, it will escape the endoplasmic reticulum quality control process. It gets secreted, but dissociates slowly into monomers, like the wild type. So the first hypothesis is saying that is a stable mutation which gets secreted into the bloodstream. Because it is stable, it does not unfold early. It takes a long time. So that's why when the patient is now old, the protein will unfold into monomers and cause disease. Now, the second hypothesis we have is that the A97S variant is unstable. So because it is unstable, it is trapped by the endoplasmic reticulum quality control system. So it does not get secreted very well. So what it does is it secretes very minimally. So you have very small secretion. Now, because you have very small secretion in the bloodstream, it takes a long time for this small concentration to build up to disease. That is why it causes a late onset disease, like we see in another mutation, which is the D18G variant. So these were the two hypotheses we have going into this project. So what we did was to understand this mutation. Like in science, we created different mutations of the disease. We created the T119M variant. This variant does not cause disease. It is very stable and it secretes in the normal secretion parts. Then we have the wild type protein, which causes a late onset disease and also secretes normally. As a as a negative control, we have the V30M mutation, which causes an early onset disease and also secretes normally. Now, we also created the D18G mutation, which causes a late onset disease. This is a very unstable mutation and it secretes very low. Now, we want to understand how our A97S performs. It is a late onset disease, but we don't know how does it secrete. So we created all these different variants. Then we transfected our cells with these different variants, and we try to see the secretory pattern and intercellular trafficking of this variant. So because the TTR protein is produced in the liver, so we use the FG2 cell line, which is the liver cell line, and we put in all the different variants and also have a mock control, which we did not transfect. Now, we transferred all this into the cell line and we'll look at the secretion in the cellular medium. So this is the negative control, which is the mock. You can see there's no secretion in the mock control because there was no transfection. Then we can see the T119M secretes very well. The wild type also secretes well. The early onset mutation V30M secretes. Now, our A97S also secretes normally like a wild type. This is our negative control. D18G is secretes very minimally and is very unstable. That's why in the cell lysate, we have very few in the cell. And this is the histogram showing the secretion and the relative secretion to wild type. So the A97S TTR mutation secrete like wild type in cells. So it's behaving like the wild type protein. Then we also try to see within the secretory pattern. So in the secretory pattern, like I said earlier, the secretory protein will be packaged in the ER. Then from the ER, they are moved into the Golgi apparatus before they get secreted out into the bloodstream. So we'll try to see how does our A97S also perform within the intercellular trafficking pathway. So for the ER, we use the KDEL and calreticulin biomarkers, which are markers of endoplasmic reticulum. Then for the Golgi, we stain with the GM130, which actually stains the cis Golgi. So if you look at the wild type protein, it's traffic within the ER, same. And if you look at the Golgi, it's actually well packaged within the Golgi. You can see the beautiful localization between the wild type protein and the GM130 here. So the same for the T119M, the V30M, and also our A97S. It was traffic within the ER and also have a good localization with the Golgi marker. So it's well trafficked within the Golgi. Unlike the D18G, which is the unstable mutation, if you look at it with the Golgi marker, you don't have that refined localization with the Golgi marker. So this picture is telling us that the A97S is well trafficked within the secretory pattern, within the secretory pathway, just like the wild type. And that is why we saw earlier it's well secreted into the medium like the wild type protein. So now to look at our variant, how stable is this A97S variant? Is it stable? So we did what we call the thermal denaturization. So this is using high temperature to unfold the protein into individual monomers. So we're trying to look at what temperature does it unfold. But like I said, the T119M variant is a very stable mutation. So you need about 102 degrees Celsius to unfold it into monomers. The wild type protein is also stable. It is about 101 degrees Celsius to unfold. Now, if you look at our A97S mutation, it unfolds at about 98.5. That's also very stable. So, and like the V30M mutation also is stable, but a little bit less stable than the wild type and A97S. So, from this chemical denaturation is showing us that the A97S mutation is as stable as a wild type, and it needs about 98 degrees Celsius to actually unfold it into the individual monomers. Now, so far, what our data is telling us is the A97S is stable, it secretes like a wild type, and it unfolds at a higher temperature, just like the wild type protein. But also, the cytotoxicity of TTR has been linked to the ability of the native tetramer to unfold into monomers. And the theory is that the wild type TTR are least toxic because they are less likely to populate the misfolded monomer pool. So because the wild type protein is stable, it does not unfold easily into monomers. So that's why it doesn't cause disease early. So any mutation that cause early disease, they unfold quickly into monomers, and these monomers are the ones that actually cause toxicity on the cells. So we try to also see how does our A97S variant perform um, in cell lines. So we made different proteins, like these are purified proteins. So we created the T1IM, which is the stable mutation. We have our wild type mutation. We have the early onset V30M and the late onset E97S. So we threw all these purified protein into the IMR32 cell line. These are neuroblastoma cell lines. We use neuroblastoma cell lines because, like I said, this disease causes FAP. So this is like a neuronal cells. So we use two different mutations. The 0.44 milligram per mil is this physiological level of TTR within the bloodstream. So when we throw it into the cell, we could see that the early onset V30M variant actually kill the cells more. You have about 60% variable cells at about at the physiological range, whereas the A97S also cause toxicity, but is not as early as the V30M and is similar to the wild type. And when we look at the LDH release is the amount of LDH that is being released when a cell is undergoing cytotoxicity. We can see that the V30M mutation, which is the early onset, released more LDH in cells than the A97S compared to the wild type and the stable mutation. So from this data, we could see that actually the A97S variant is less toxic than the early onset V30M mutation in cell lines. So we want to link all our data to the previous stability theory. So the stability theory tells us that the liver produces this TTR protein. Now, the TTR protein, which is a tetramer, has to unfold into monomers. And like I said earlier, this unfolding is the rate limiting step for the disease to occur. Now, when this TTR unfold from tetramer to monomers, now these monomers will now unfold into amyloid fibrils. And the amyloid fibrils are the one that deposit in different organs and cause disease. Now, how does our late onset A97S variant perform so far? In terms of secretion and escaping the ER quality control, what we saw was that the A97S mutation, like other moderately stabilized variants, is secreted like the wild type. It's kind of stable like the wild type protein. And in terms of cytotoxicity, it is mildly toxic than the early onset V30M mutation. So that means it is less likely to populate the misfolded monomer pool. So that is why it might maybe cause the late onset disease. But the question we ask ourselves is this stability the only reason why our mutation is causing a late onset phenotype? And the reason for this question is because the stability theory has some misfit in it. It doesn't fit in all scenarios. Now, let me give an example. Now, the wild type protein, which is the control, is secreted normally within the cells into the bloodstream. It has a dynamic stability of 3.4. That means it is stable. The tetramer monomer pool is stable. Now, it has a kinetic stability of 41 hours. That means before you to unfold it into monomers, you need about 41 hours for the tetramer to unfold into monomers. That's a long time, and it causes a late onset disease. Now, if you use this model to look at other mutations like the L55P mutation, this has a normal secretion in cells. It has a lesser thermodynamic stability. But this is interesting because if you look at the kinetic stability, it takes about 4 hours to unfold. So if it unfolds early, most likely it's going to cause an early onset disease, and which is true. This L55P causes an early onset disease. So this fits the model of stability. But the common V30M mutation and the V122L mutation do not fit this stability model. How? Because the V30M mutation secretes normally. It has a lower thermodynamic stability. Now, similarly to wild type, it takes about 40 hours to unfold from tetramer to monomers. But despite that, it still causes an early onset disease. So that means it does not fit the stability model. Then, lastly, the V122I mutation secretly normally like the wild type protein has the thermodynamic stability of 3.4, similar to the wild type. Now, interestingly, it takes about 19 hours to unfold. So if it takes 19 hours to unfold, we expect it to cause an early onset disease. But no, this causes a late onset disease. So this also does not fit in the stability model. So like I said earlier, there are misfits in different mutation to the earlier stability model, and not all mutation fit into this model. So that was why we asked the question, is the stability of the A97 STTR responsible solely for the late onset phenotype? Now, to address this question, what we did was to we dig deeper into the literature, and there's this classic paper that was published in EMBO journal in 2007. What it says is that there's non-disease causing monomeric TTR can escape the ER. So that means when you have the mutations that do not cause disease, when you create their monomers, they actually can escape the quality control of the ER, and they get secreted as into the bloodstream. So if you look at this data, this is the cell lysates. They have the wild type, and these are two stable mutations which don't cause disease. These are non-amyloidogenic, don't cause disease. And if you look at it, they get secreted into the medium. And the paper actually showed that the disease-forming mutations, they are trapped by the endoplasmic reticulum quality control. So they used different disease-causing mutations like the D18G, H25T, V30M, E55K, and L55P. When you put the monomers into the cell, you can see that they don't get secreted. They are trapped by the ER quality control. So the monomers of disease-causing TTR mutation do not get past the ER quality control. So that was what this paper said in 2007. So the question we ask is this true for A97S monomers? What if let's see, how does it fit into this publication or this data? So what I did was to create the monomer of the A97S, and to our surprise, the A97S which is a disease-forming mutation, actually the monomer can bypass the ER quality control. So if you look at this data, this is the V30M early onset. This is our A97S mutation. This is the non-D18G, and this is stable mutation. So if I go back to the data here, you can see the T119M here gets secreted, and the V30M and the D18G do not get secreted, and they are disease-causing mutation. But in the case of A97S, which is also a disease-causing mutation, it gets secreted into the medium. But the V30M does not get secreted. The D18 gene does not secreted. So that means our A97S, which is a disease-forming mutation, actually can bypass the ER quality control, which is something different from what the previous literature have said. Now, to get a more clarification, we look at our data, that what is happening. So what we saw was that the previous data, the A25T, V30M, E5K, and L55P, they are all early onset disease causing mutation. And our V30, our A97S is a late onset disease mutation. That means, is it that all late onset mutations can bypass the quality control in the monomer stage, while the early onset do not? So to answer this question, what I did was to look at the different mutations in the TTR. Like I said, the TTR has about 100 different mutations, and it spans exon 2, 3, and 4. So at each exons, I pick two late onset mutations, and I create their monomeric variant to see how they actually behave within the cell. So this was the mutation I picked. So within exon 2, I picked P24S and A45G. These are late onsets. And exon 3, I picked F64L, H88R. In exon 3, they're also late onset. And in exon 4, I picked I107V and V122L, which is also a late onset mutation in exon 4, just to rule out exon-specific differences. Now, I created all these mutations in the monomer stage to see how they behave within the cell. Interestingly, when we put all these late onset monomers within the cell, they all bypass the ER quality control. So you can see all of them they are secreted except the early onset V30M and the D18G, which is the highly unstable mutation. So all late onset monomers actually can bypass the ER quality control. So from this data, we propose a new mechanism for the disease onset, which is different from the stability theory that was proposed earlier. So this is what our theory says. Our theory says for early onset disease, like the V30M, within the ER, they style as folded monomers. Now, because they are early disease forming mutations, they cannot secrete as monomers. They are trapped. So what happens is they have to come back. The monomers will form tetramers. Now, these tetramers are now being secreted into the bloodstream. Now, because the tetramers are unstable, they unfold into individual monomers, and these monomers will misfold into aggregates and they cause early disease because of stability. Now, for the late onset disease, like the A97S, within the ER, they start as folded monomers, and these folded monomers can also come together as tetramers. Now, because the tetramers can get secreted and they are stable, so their disease is not due to the unfolding of their stable tetramers. What happens is their folded monomers can also escape the ER quality control, but they escape in very low concentration. So it takes a longer time, years, to achieve concentration which is enough to cause disease. Then that's when they misfold into the amyloid and they cause late onset disease. So we kind of brought a new mechanism in which the cellular um, processes is what predicts the onset of disease, either early onset or late onset, not necessarily the stability of the tetrameric form of the mutation. Now, after doing this, the next thing we did was to um, model this mutation in fly, and like FIRAT is also is proposing we do research with Drosophila mellitus, which is a very good model. And the reason why we model in fly is because it's been very difficult to model this disease in mice. So, and different groups have reported the successful generation of Drosophila model for ATTR FAP. So we collaborated with Dr. Chan at National Taiwan University, who created for us the HTTR fly models. Now, the first thing we did was to have like the normal Drosophila genetics here. You have the US line, which has your protein of interest, and you're going to cause this US line, which is your GAL4 line, which is to drive your protein of interest in different area of the fly. So when you cause it with the D42 GAL4, you're going to be expressing your proteins in the motor neurons. If you use the NCIP-GAL4, you're going to be expressing your proteins in the nervous system. So for our study, I use both the D42 GAL4 to run the proteins into the motor neurons, and I assayed for the choline acid and neuromuscular junction morphology. Then in adult fly, I caused the US line with the NCIP-GAL4 to look at the climate assay and the lifespan assay for this. So for here, we created the wild type fly, the early onset V30M variant, and also our A97S variant, which is the late onset but very aggressive disease forming variant. Now, how do we know that our flies are actually working? So the first thing we have to do is to make sure our fly actually can produce the protein. So when you cross the US GAL4 lines to the NCIP GAL4, you are running the protein in the nervous system. So I took out the brain of the fly and I did the Western blot to see if they're actually expressing the protein. So if you look at these blots, the NCIP GAL4 doesn't have the TTR protein. So that's why you don't see any TTR protein here, because obviously we did not cause it with any TTR line. But the wild type beta-TM and A97S all has the TTR protein. So this is telling us that our fly model actually works. We can actually deliver our protein in different organs or different part of the fly from this data. So the first thing we did was this is a video, but I don't think it's going to play. Yeah, it's not playing. So the first thing we did was to look at how does the TTR variants perform in the larvae of the fly. So what we did was to cross the GAL4 lines into the D42. The D42, like I said earlier, is going to express the protein in the motor neurons. So when the flies are in the larvae stage, we try to see how does the movement change? Does the protein actually affect their movement? So if you look at this, we can see that the V30M early onset actually starts having reduced movements in larvae, which correlate to the early onset disease of this fly, of this model. So that means as early as in the larvae stage of the fly, it's in the motor neurons, is actually affecting the motor neurons, and they are not actually moving well compared to the late onset E97S and the wild type and the D42, which doesn't have any protein expressed. So the V30M actually have reduced calling in the early stage in the larvae. Now, we also look at the neuromuscular junction of the larvae just to see do the do these proteins actually affect the neuromuscular buttons? Why is that the reason we are seeing reduced movement? But when we look at the neuromuscular junction, this is staining the neuromuscular junction of the larvae using the HRPDLG stain. We couldn't see any difference in the number of buttons of the neuromuscular junction. So that means the early onset reduced calling that we see in the V30M is not due to the destruction of the neuromuscular junction, but just because of a different way the protein is impacting the the whole movement process of the fly. Now we went to the adult fly to see how does mutation upon how does it affect the adult fly. For people that work on flies, the climbing assay is one of the assay that is used to look at decline in neuronal functions in fly. So also this is a video, but it's not showing here. So we have the NC. So for negative control, we use an amyloid fly, which is a Alzheimer's fly, A beta 42 fly, caused to the NC GAL4. Then we have our late onset A97S in blue, the early onset V30M in green, the wild type in red, and the control NC GAL4 in black. So as early as 7 days as adults, we could see that the the Alzheimer's fly actually performing very weakly, and by 14 days, none of the fly can fly. But for the early onset V30M, as early as 7 days, they are being impacted. They can't climb. They can't climb the tubes, and by 14 days, you can very see little of them climbing, and by 21 days, little of them can climb. But the V30M, the A97S, just like the wild type, at 7 days they can still climb the tube. But for our A97S, by 14 days, because it's a very aggressive mutation, by 14 days, when the disease has set in, the flies could not climb, and by 21 days, they cannot climb any longer. So we use this Drosophila model to demonstrate the early progressive climate defects in the V30M fly, and also the late fast progressive decline in the A97S flies. Now, we now finally look at the how does the mutation actually affect the lifespan of the fly. So we did the lifespan assay in fly, and the negative control in the Alzheimer's fly has about 21 days of lifespan when you put them in 37 degrees and look at the assay. By 21 days, most of the flies have died are dead, whereas our A97S, because it's a very aggressive disease, by the time the disease sets in, the lifespan is about 40 days, which is lesser than also the early onset and the wild type um, flies, whereas the NCIP-Gal4 doesn't have the mutation, can go as early as 60 days. So what is it that is showing us that by the time the disease sets in, the A97S fly is very aggressive and actually have a reduced lifespan than the early onset B30M flies. Now, so what we concluded from our study, from the fly study, is that we could use the fly to model the A97S patient phenotype, and we could show the late onset disease using the climate assay and the aggressiveness of the disease progression using the lifespan assay. Now, going to the second part of my talk. So having done this biomedical research and publishing papers, so why did I switch to a clinical fellowship? I think this is where I'm going to spend most of the time, the remaining time I talk. Why did I go into clinical chemistry? As someone that studied in neuroscience and you've done research, you've published, why did you move into clinical fellowship? Now, for every PhD student out there, when we're starting our PhD fellowship, when we're talking with our advisor, when the advisor asks us, what's your plan in the future? All of us usually say, I'm going to be a professor at a major research university after I graduate. We all want to be professors. We all want to have our own labs. We all want to do research. That is the reason why we did a PhD. And starting the PhD, that's in our mindset. That's okay. By the time I finish my PhD, I'm going to go ahead and become a professor and have my own lab. But in the real world, most PhDs actually have to look for career alternatives because not all of them actually become research professors. Now, this is one of the reasons I moved to the clinical field. So because at the time I was finishing my PhD, I saw a paper that was published in Nature, and this is like the picture from the paper, something similar. So what it's saying is that 89% of PhDs want to be professors. And I think this is still true when you ask all PhDs, what do you want to do after your PhD? They say they want to become research professors. Now, most of the PhDs go ahead and become postdocs, and about 60% of postdocs in life sciences do more than one postdocs. And if when you look at statistics, 83% of all postdocs want to be professors. But in reality, only 10% of PhDs will eventually become research professors. So the remaining 79% actually have to look for career alternatives out of research. So what are the alternatives apart from research? So there are different clinical fellowships which PhDs can also do, which you don't necessarily have to do with basic research. So you can leverage on your PhD and do different other fellowships that can give you some avenue to also do research, but it's going to be in the clinical side, and you can still publish. And some also come with assistant professor roles. So I put this for awareness. So we have 4 or 5 different type of clinical fellowship for PhDs. One is the clinical chemistry, which I did, and this is accredited by the Commission on Accreditation in Clinical Chemistry or by the Canadian Academy for Clinical Biochemistry. This fellowship takes 2 years to do. When you finish this fellowship, you become a clinical chemist and you can do clinical research and do different things like what I'm doing now. We also have the Laboratory Genetics and Genomics Fellowship and also the Biochemical Genetics Fellowship. These are both regulated by the American Board for Medical Genetics and Genomics, or the Canadian College for Medical Genetics. So this fellowship takes 2 to 3 years to finish. And when you finish this fellowship, you become a clinical laboratory geneticist or a biochemical geneticist. And you can go ahead, do research in the field and also publish in the field, and you also work in the healthcare sector. The other fellowship that is there is the HLE fellowship, which is the Histocompatibility and Immunogenetics Fellowship. And this is regulated by the American College for histocompatibility and immunogenetics. Now, previously, this fellowship takes 2 years, but now, from last year, they make a new rule, so it's going to take 4 years to do this fellowship. And when you finish this fellowship, you become an HLA lab director. Then the last fellowship that is available for PhDs are the microbiology and also the immunology fellowship. This both of them are regulated by they're accredited by the American Society for Microbiology, and this fellowship takes 2 years to complete. So now, when you do this fellowship, I'm going to go into each of them to tell you what you do or what this fellowship is preparing you to become when you do them. So now, let's start with the clinical chemistry fellowship. So the objective of the fellowship is to train individuals to manage or direct high complexity laboratories so that you become clinically oriented. I can also participate in a variety of translational research projects. So when you undergo a clinical chemistry fellowship, basically they are training you to become a director of a clinical chemistry lab. And a clinical chemistry lab is one of the biggest labs in the world because when someone gets sick, you go to a doctor, the doctor asks to do some blood tests. You go to the clinical chemistry lab, they get your blood, and the blood is being run in the lab. So it is your duty as a clinical chemistry lab director to ensure that the test is done adequately and the results are compatible and reliable for clinical diagnosis. So what we do basically in the clinical chemistry lab is just basically three things. We have the pre-analytical side, we have the analytical side, which we do in the lab, and we have the post-analytical side. So, like I said earlier, when a patient is sick or you go to a doctor, they give you a sample, your blood sample. The blood sample is being taken and sent to the lab. So this is where you come in as a clinical lab director. You ensure that the lab tests the blood sample and the results are reliable by making sure the quality control processes are being followed and the result is fine. Then the result is sent from your lab to the ordering physician who uses this lab results to diagnose and monitor or treat the patient. So this fellowship is 2 years. It gives you everything you need to perform this role. And by the time you finish the fellowship, you can become a lab director that directs a lab like this as a PhD. Now, the second fellowship I discussed was the Laboratory Genetics and Genomics Fellowship, or the Clinical Biochemical Genetics Fellowship. So, the aim of this fellowship is to train individuals to be proficient in directing and interpreting both clinical cytogenetics and molecular genetic analysis essential for diagnosing and managing human genetic diseases. So, when you do this fellowship, which is either 2 or 3 years, depending on where you do the fellowship, they are going to equip you with what you need to be able to interpret clinical genetics data to diagnose and manage patients that have genetic diseases. So, like in this part of the world, we have a lot of disease-causing mutations, lot of so in which you need to diagnose them early so that the doctors can know, okay, this patient has this mutation in this gene. We have to do maybe um, some genetic treatments, or you have to ensure that this patient doesn't marry this kind of partner so as not to have children that have the kind of disease. So this is just going to train you to become to become a director of this lab. Now, the other fellowship I talked about was the HLA fellowship. So the HLA fellowship is um, include that do the transplants, so HLA transplants lab. So when you do this fellowship, it's going to provide you with technical training in the field of histocompatibility testing and transplant immunology, so that you'll be equipped in managing a transplant center. So when you can work in a transplant center where they do kidney transplants, heart transplant, or any major solid organ transplant or bone marrow transplant. What you are doing as a director is to make sure that both the donor and the recipients are compatible. They are compatible. There are no HLA antigens that can destroy the organ or destroy the bone marrow after they've been transplanted. So you as a PhD, you can do this training and you can eventually direct a lab like this to impact patient care, ensuring that transplantation are compatible between donors and recipients, just so as to prevent graft rejections or failure in this patient. Now, the last one is the microbiology or the immunology fellowship. So, this fellowship will prepare you to become laboratory directors of medical or public health laboratory that focuses on microbiology and medical immunology. And by the time you finish this fellowship, you'll be able to find work in a clinical microbiology or immunology laboratory or at academic center or hospital setting. So basically, just like the chemistry and the genetics, you'll be a lab director of microbiology, and you'll make sure that all the microbiology testing that is being done is being done well. The results are of quality, and the doctors or physician they can rely on your result to diagnose or manage their patients adequately. So all these four fellowships are out there for PhDs to do, which are like alternatives to just doing the biomedical research or getting stuck as postdoc for long years. Now, this is the end of my talk. This is the most important slide of this presentation. I have to acknowledge Professor Adedeji. Um, he has been a mentor for a long time, since over 12 years now. He has been my mentor, and I know he's been doing a lot of work to ensure research in Africa is being done well and to give young scientists the platform to learn more about research and do well. So I want to acknowledge Professor Lee for all the support he has given for over the years. I'm also very grateful for FIRAT for giving me the opportunity to give this talk. I hope I'm able to inspire people. And if there's any question on how to get into all this fellowship, I'm happy to answer. I also want to thank Dr. Folarin for inviting me to give this talk. And I'll thank my previous lab, PhD lab, Professor Jim with Science Lab, where I did the first part of the work for the amyloid disease study. Thank you so much for your attention. I will take questions, comments at this point. Thank you very much.
Thank you very much, Dr. Ridwan Ibrahim. I must say that we've learned a lot from the lecture you give us, particularly the second aspect of your work. Thank you very much for giving us this for creating time to give us this talk. So we are looking at questions from audience so that we can give an insight to the questions raised.
Yes. So should I read the question from the screen? I can see some of them popping up now.
Yes, please. Go ahead,
Yeah.
sir.
Yeah. The first one says, have any studies looked at the effect of alternate splicing on misfolding of TTR? As at the time I did this study, there was no. Actually, I was trying to look at this, but as a PhD student, you just want to do your work, publish, and leave. So at the time I did the study, there was no study looking at the alternate splicing. But for now, I've not gone back to check the literature. I'm sure there will be studies now which have looked at how alternate splicing actually could be one of the reasons to that could predict onset of disease. Yeah. But when I did this study, there was not any work on alternate splicing. And as a PhD student, I didn't want to just spend more time. I want to graduate. I already spent 5 years, so I just publish my studies and leave it for other people to carry on. In research, you can't do all the studies. You do your part. There will still be unanswered questions for other people to come back and also work on. That's a question from Michael. Next question. Yes, could chaperone play a role in early only? Yes. So, like I talked about, endoplasmic reticulum assistant folding. So chaperones actually help in this folding pass, and there have been a lot of chaperones that has been implicated at HSP protein. They help to package moderately stabilized TTR variants to fold them because they can still be folded. So they'll be folded in the ER, and because before they go to the Golgi and get secreted out. So chaperone actually plays a huge role in stabilizing the protein within the ER. So then like I said earlier, when the initial model was saying that when the chaperones helped within the ER to package them into folded tetramers, when they get secreted into the cell, into the medium, then their stability now predicts whether they become early onset or late onset now. Well, the chaperone actually help in the folding process. No, so could we classify TTR as a prion protein? No, TTR is not a prion protein because it's not a transmittable. Prion proteins are transmittable diseases like the prion disease that we know. So TTR is actually it's a protein inherent in the body. It has its own mechanism of unfolding into amyloid fibrils and depositing. So it's not a prion protein. It's a secretory protein. Are they all the questions, Dr. DM, or we still have more coming?
I think that's all from the background here. I think that's all.
Okay. If there's any question on the clinical part, also I'm happy to answer. Then, if not, I will leave the stage for the organizers.
Thank you very much. I was going to ask that because consider the fact that for some people that study their PhD in here in Africa, there's that challenge with not really looking at or kind of looking down on the PhD from Africa. Wouldn't that be a challenge considering people that are willing to consider that option you said, sir?
Yes, thank you so much. That's a very good question. So, a PhD is a PhD. Nobody is going to look down on a PhD. So, especially when you meet up with the requirements and you actually you can show that you have the zeal. Though this fellowship, I must admit, are very competitive, very competitive. It took me my second trial of applications to get in. It's very competitive, but getting the PhD from Africa, it does not write you off for getting the fellowship. So the most important thing is making sure you have the requirements and you can demonstrate in your SOP your zeal to improve patient care, and definitely you stand a higher chance of getting to the fellowship programs. So it's not about where you get a PhD from, although because of the world is working right now, in terms of visas and everything, that could be a restriction. So that's why when I finished my PhD in Taiwan, I had to move to Canada to be closer to where the fellowship is being done. For example, like the Canadian programs, they give preference to people who are Canadians or Canadian permanent residents first. So that could be one of the reasons why you want to come to this part of the world to meet that requirement. But for the US, it doesn't matter. We have a lot of people that are being brought from Asia, from different places. It's just the ability to demonstrate um, that you meet up the requirements and you show the zeal of advancing patient care, and you stand a high chance of getting the fellowship program. It doesn't matter where you get a PhD from. Now, I saw a question that says, what is the process of applying for this fellowship and what are the requirements? So each of the fellowship program has their unique requirements. Like, for example, microbiology, you must have a PhD in microbiology or immunology. For the HLA, you have to have a PhD in life sciences or immunology. For chemistry, you can have a PhD in life sciences, or if you have a PhD in chemistry or analytical chemistry, you stand a higher chance. For the genetics, a PhD in life science or genetics is very encouraged. Or if you have a research that has to do with genetics, you stand a higher chance of getting in. And the process of applying differs. So they all have a deadline for applying. So each of the program, like I put in the slide, when you go to the names of the bodies that are accrediting this program, they have a list of all the places they do these fellowship programs. Each of them has a different date for applying, a date for reviewing, and date for interviews. So for anybody who is interested in applying, I will ask them to like kind of get an Excel sheet of all the programs. Go to the individual website of the program, look at the dates of application. So that they all have different dates. So you know this is a date for each of the program. Then you apply it to them using your SOP and tailor your SOPs to the description of the program. So for example, I train in Texas Children's Hospital. We focus more on pediatrics clinical chemistry. So in my SOPs, I make sure I'll show that like I'm interested in pediatric chemistry. But if I'm applying to a program that's an adult chemistry lab, I'll tell you, okay, I'm interested in adult clinical chemistry, just to you are kind of tailor your SOP to the description of the program, and with that you stand a higher chance of getting the fellowship programs.
Thank you very much, sir. Um, I must say that we've learned a lot from your talk this evening here in Rwanda. So I hope that, having said that, I hope that when next we call upon you to share with us from your wealth of experience, you will not hesitate. Um, you would openly and graciously open your two arms to accept our invite. I must say that we still have a lot to learn from you. Thank you very much.
Thank you. Anytime, I'm always willing to share anything I know, and always willing to give back to my people. So anytime you call on me, I'm willing to do that. Thank you so much for the invitation.
You're welcome, sir. So to the people at the back end, thank you very much for the hard work you are putting into this program. Without the efforts from the MD to the IT, Mr. Abdulaziz, this wouldn't have been a reality. Thank you very much. And also to the audience who have taken their time to listen to our guest lecturer. Thank you very much. I hope that we must have learned one or two things that we would have to take back to our different scientific communities and relate to them the message or the gospel from um, Dr. Ridwan Ibrahim. Um, till we meet. Okay, I think we have announcements. As you can see from the work of the guest lecturer, you know, in his work they have used the fly model to do their work. That's why we are also trying to establish a Drosophila lab here in Rwanda, as well as to train people so that we can also do in-depth analysis, just like the presenter has presented in his work. So we intended to train people, but unfortunately, due to some challenges, we will not be able to do that at the moment. So please be on the lookout for the call next year, God willing, for the training on Drosophila here in Rwanda. So till we meet again, see you when we see you. Thank you very much. Bye.
As researchers, we share something rare, a commitment not only to knowledge, but to the responsibility that comes with discovering it. Every study, every design choice, every participant interaction, all of it represents an investment of thought, effort, and often personal sacrifice. Late nights preparing protocols, revising instruments after feedback, balancing teaching responsibilities, publication goals, and grant deadlines. And through it all, you keep going, not because it's easy, but because the work matters. Yet, in conversations with researchers across disciplines, across continents, we hear the same quiet truth expressed again and again, that the administrative weight of research has grown heavier than many of us ever expected. Not the science, not the analysis, but the thousand small tasks that fill the gaps between ideas and results. Formatting, reformatting, fixing logic issues, troubleshooting participant feedback about an interface you did not design, preparing clean exports for statistical software, going back and forth with IRB committees over platform limitations you didn't create. None of these tasks diminish the integrity of your work, but they do steal time from the work only you can do, the deep thinking, the interpretation, the mentorship, the creative reasoning that turns data into insight and insight into impact. And then there's the part we rarely speak about openly, the participant experience. Participants who want to help, who believe in the work, but who find themselves navigating forms that feel overwhelming, dated, or unintuitive. They're not disengaging from you, they're disengaging from a process that doesn't respect their cognitive load, their device limitations, or the realities of their day-to-day lives. And when participants struggle, data quality suffers, completion rates drop, researchers work harder, not smarter, trying to rescue datasets that should have been strong from the start. For years, our field has had limited choices, tools that are easy but lack rigor, or tools that are powerful but cumbersome, costly, and draining. But compromise should not be a prerequisite for discovery. And research tools should honor, not hinder the work researchers dedicate their lives to. So today, on behalf of the Foresight Institute of Research and Translation, I am grateful to introduce something new, a platform designed around the lived realities of researchers, built not to replace your expertise, but to support it with clarity, efficiency, and respect. It's called Snap Genius. Snap Genius was created for one purpose: to give you back the time you've lost to administrative friction and to return your focus to the intellectual work that drew you to research in the first place. You describe your study in a single sentence, and a complete, structured, logic-ready form is ready in under a minute. Consent forms are formatted correctly, field types are aligned to best practices. Design is modern, mobile responsive, and participant-friendly. Participants move through studies one thoughtful question at a time. No clutter, no scrolling, no cognitive overload. A clear, calm experience that respects their time and in return protects your data quality. Audio diaries, digital signatures, and follow-up scheduling are fully integrated. Data collection in environments without internet is seamless. Responses are stored securely and synced automatically when a connection returns. After data collection, Snap Genius transforms the analytical experience. You can query your data directly, run correlations, generate visualizations, and perform statistical analysis without leaving the platform. Literature reviews are summarized intelligently, citations formatted automatically, and method sections drafted from your actual study structure. Security and privacy are foundational. End-to-end encryption, GDPR and CCPA compliance protect your data. No ad tracking, no selling participant information, full control remains with you and your participants, giving IRBs and collaborators confidence. Snap Genius is also cost-effective. Legacy platforms can cost over $1,500 per year. Snap Genius provides full functionality for a fraction of that cost, freeing your budget for participants, equipment, or other critical research needs. Your research deserves tools that elevate your work rather than exhaust it. Snap Genius restores efficiency, improves participant experience, and returns time for the intellectual tasks only you can do. Thank you for the work you do and for the discoveries you pursue. I'm Mira Wurst. On behalf of the Foresight Institute of Research and Translation, I invite you to begin your complimentary month of Snap Genius today. Test it with your own projects. See how it changes the pace and quality of your workflow. If it doesn't free your time, reduce administrative burden, or improve participant experience, simply let it go. But if it does, if it gives back even a few hours each week, or clarifies your analysis, or improves data quality, we would be honored to support you as you continue your work. Your ideas deserve that support. Your time deserves to be protected. Your research deserves tools that rise to the level of your commitment. Click the link now to start your complimentary month of Snap Genius. We look forward to supporting your next discovery.